Now let's take a look at the lymphatic vascular system. Lymphatic vesicles convey fluid called lymph throughout the body. Lymphatic circulation is unidirectional from the tissues to the venous circulation. It consists of two types of vessels, lymphatic capillaries, which include lacteals, and lymphatic vesicles from small to large. The largest lymphatic vesicles are the right lymphatic trunk and the thoracic duct. Lymphatic capillaries feature blind ends. They are simple endothelial tubes, and they are kept in place by anchoring filaments, which have some elastic components to them, but really are for structural support to keep those otherwise unsupported walls open. They have a discontinuous basal lamina, and this feature, along with their very thin walls, allows tissue fluid to get into them and proteins that are too large to traverse into the blood vasculature. Also, a lot of lipids can travel through this system very easily as well. In fact, large proteins and lipids often enter the body after digestion and transcytosis. They enter into lacteals, which are lymphatic capillaries found inside the villi in the gastrointestinal tract. And you can see a couple of examples here. So both at the light microscopic and electron microscopic levels, a couple of features are apparent in these lymphatic capillaries. They have very, very thin walls, which consist of really just a simple endothelial tube. We call that very thin wall attenuated. In other words, it's stretched out. They also have a very angular appearance because they're being held open by those anchoring filaments that pull the walls outward. Small to medium lymphatic vessels can be found in many locations, and they are often conduits for lymphocytes, as you can see in this example. Medium lymphatics begin to have more than just a simple endothelial wall, and you can often see a layer of smooth muscle, as in this example. Sometimes lymphatics, as they become larger and larger, have valves, and you can see an example of a valve here in this micrograph. Note the very thin wall of that lymphatic in comparison to the adjacent artery. I guess it looks like a small muscular artery next to it. Large lymphatic vessels, such as the thoracic duct, have features similar to a vein, but they're not as well organized. In other words, those tunics are very indistinct. In other words, it's hard to tell where the tunica intima ends and the tunica media starts, and sometimes even it can be difficult to see where the tunica media and the tunica adventitia are separated from one another. In this example, you can really see the tunica media pretty clearly. It's the dark pink line directly around the lumen. And then outside that, you have the collagenous connective tissue of the tunica adventitia. Sometimes they have circular and longitudinal smooth muscle, similar to the larger veins, but again, it will be very poorly organized, and it just will not look as well put together as a comparably sized vein. Now let's take a closer look at the cells that line all of these systems, the endothelial cells. They structurally are usually elongated parallel to the direction of blood flow, and as mentioned previously, they're almost always attached to one another by a variety of junctions. They are anchored to their underlying basal lamina by hemidesmosomes, because essentially they're epithelial cells, just like the epithelial cells we studied way back in Foundations of Medicine. Now, what's different about them from other epithelial cells? They contain a specialized organelle called a Weibel-Palade body. So Weibel-Palade bodies contains something very important. It's called von Willebrand factor, and that plays a major role in blood coagulation. Deficiency of this factor results in von Willebrand disease. So what other things do endothelial cells do? Well, they have many functions. They maintain that selective permeability barrier between the vasculature contents and the underlying tissue compartments. They maintain non-thrombogenic barrier between platelets and the subendothelial tissue. They modulate blood flow and vascular resistance. They have a regulatory role in cell growth. In other words, they secrete growth factors. They participate in the regulation of immune responses. They help maintain their own matrix, the basal lamina and the glycocalyx on the luminal side. They inactivate and convert compounds in the blood, most specifically angiotensin 1 to angiotensin 2. It's a crucial function of theirs. And they act in the modification of lipoproteins. So there are a number of conditions that are important clinically related to the structure of the vasculature. First of all, we have aneurysms. This is where you have a weakening in the vessel wall, usually related to the tunica media and a specific defect in collagen. You have ischemic heart disease, typically caused by coronary artery obstruction. This leads to diminished blood flow to the myocardium and can result in angina and can even progress to MI, myocardial infarction. Another common condition is varicose veins. This is where the veins become abnormally dilated due to an alteration in the vessel wall and sometimes valvular incompetence in the veins that prevent backflow. You can also have conditions related to transplantation of organs. In this case, there is no autonomic nervous system innervation that modulates and coordinates the organ within the entire environment. Finally, we have to take a quick look at atherosclerosis. This is an extremely common condition. It's being found these days even in young people. So it used to be thought of as a senescent disease that accumulates over time, but really even young people with poor diet and exercise profiles can have atherosclerotic plaques. So what's happening here? These are specific lesions in the vessel wall called atheromas. And what happens here is that within the tunica intima, the most internal layer of the vessel, there's an uptake of monocytes into this region of the vessel wall, along with certain growth factors like PDGF. The PDGF also stimulates smooth muscle cells in the tunica media to enter into the tunica intima where they don't normally belong. Both the macrophages and the smooth muscle cells can end up uptaking a lot of LDL because this condition is related to high cholesterol, high LDL in the blood itself. And when they uptake that, they become foam cells. In other words, they're full of the lipid. The plaque then will contain a lot of fibrous connective tissue, which is made by some of the cells that migrate into that area, because as I've said before, the smooth muscle cells can be highly synthetic. Smooth muscle cells that don't really belong there, foam cells, other macrophages, lymphocytes come in as a reaction to the area, and cellular debris from breakdown. So these atheromas can occlude the vessel. They can provide a very rough surface, which changes the vascular flow. They may, in fact, lead to myocardial infarction, which can, in some cases, be fatal. So that's all we have, and we'll see you in lab.